We are moving from the macro logistics of the blood into the microfactory of an individual cell.
And this is where the geography gets really, really interesting. So we've entered the cell, we're in the cytoplasm,
Yeah.
but the ultimate goal here is to get to the mitochondria.
But not everything happens in the mitochondria right away.
Mm-hmm.
The source points out a split here. We've got 4 major pathways to talk about.
Glycolysis, pyruvate dehydrogenase, the TCA cycle, and beta-oxidation.
And only one of those is happening out in the lobby of the cell, the cytoplasm.
That's glycolysis. It's an incredibly ancient pathway. It's happening in the cytosol, and it's handling that glucose we absorbed earlier.
And this acts as the bridge, right?
Mm-hmm.
Because glucose is a 6-carbon molecule. It's too big to just waltz into the mitochondrial machinery as is.
Right. Glycolysis literally means splitting sugar. We snap that 6-carbon glucose into 2 3-carbon molecules.
Pyruvate.
Pyruvate, and pyruvate is the ticket. That's the molecule that effectively knocks on the door of the mitochondria and says, okay, I'm ready for processing.
So glycolysis is the prep work. It's like chopping the vegetables before you put them in the oven.
That's a great analogy.
So let's talk about the oven, the mitochondria. The source material really emphasizes that the other 3 pathways, pyruvate dehydrogenase, TCA, and beta-oxidation, are all strictly mitochondrial.
They are deep inside in what's called the mitochondrial matrix, and there's a very good reason for that sequestration. These pathways are the real heavy lifters. They're stripping electrons off our food molecules.
Okay, let's break down what these 3 actually do, because the names can be a mouthful. Let's start with pyruvate dehydrogenase. We've got our pyruvate from glycolysis. It gets into the mitochondria. Then what?
Pyruvate dehydrogenase is kind of like the gatekeeper. It's a complex that takes that pyruvate, clips a carbon off it.
Which we breathe out as CO2.
What breathes it out is CO2, and it converts the remaining 2-carbon piece into acetyl-CoA. And acetyl-CoA is the universal fuel for the next step.
So it converts the raw delivery into the specific fuel type the engine can run on. What about the fats? The ones that arrived in those chylomicrons?
They go through beta-oxidation, and you can think of beta-oxidation like a salami slicer.
Salami slicer.
Yes. Fatty acids are these really long chains of carbon.
Yep.
Beta-oxidation just cycles through and chops them into 2 carbon units over and over again.
Okay.
And guess what those 2 carbon units are?
Let me guess. Acetyl-CoA.
Exactly. So whether you ate bread, which is glucose, or cheese, which is fat, the cell funnels them both toward this one central molecule, acetyl-CoA.
Now that is efficient. It standardizes the fuel source. So now we've got a big pile of acetyl-CoA sitting in the mitochondrial matrix. This feeds into the famous TCA cycle.
Or the Krebs cycle.
Right.